Hi everyone, today I'm delighted to have Professor Louise Emmett to talk about all things radiopharmaceuticals. So Louise, welcome to the show and thanks for coming on.
Thanks for asking me.
So I thought, given a lot of our listeners are investors, it'd be great to talk about PSMA and what's happening in the space. So what's the current state of the art? Like how are people treated? How are those radiotherapies used today?
So it's a massive question because PSMA has really transformed the prostate cancer landscape, both right from diagnosis up to metastatic castrate-resistant prostate cancer, from an imaging point of view and then from a therapy point of view. So we really started with therapy, and we started with staging of prostate cancer patients, biochemical recurrence of prostate cancer patients with gallium PSMA-11, and then that's expanded to diagnosis of prostate cancer patients right up to assessment for treatment response in imaging, and then that again is expanded from gallium PSMA-11 across to multiple different fluorinated agents, copper agents. So, the space is changing in terms of the sensitivity and specificity of what we're going to get, what evidence we've got for its use. And then you're talking about treatment. That is also a rapidly changing space with lots of new PSMA peptides, but also antibodies and then different radionuclides. So, we have copper-67, we have lead coming on board, we have lutetium, we have actinium. Huge field now.
Yeah, so that field seems to really have taken off in the United States, certainly in terms of revenues and product. So how's it used over there, and how does that defer to how those agents are used in Australia?
Yeah, so it's a very different landscape in Australia to the US. First of all, when we talk about imaging, in Australia we have utilised GLP compliant laws to really expand the use of gallium PSMA-11, which is an off-peptide agent. So gallium PSMA-11, rapid introduction to clinical practice in Australia because it's cheaper. It's about $750 to get a PSMA PET scan in Australia before it was Medicare rebated, as opposed to about $7,000 to $15,000 in the States to get the same one. So the indications for use are very similar in the two countries. It's much harder to get a PET scan in the US than it is in Australia. But it's much more lucrative to the companies to provide those PET scans because of the GMP compliant laws in the US. You can't do in the US what we do in Australia, which is local hospital production.
Right. Why is that?
It's historical. I think we're a very small country where, in terms of population-wise, what does Oliver Sartor call us? He calls us a pimple. And, I prefer to say that we're a freckle. But so what the federal government did many years ago to try and get access to a lot of these scans in nuclear medicine without them having to be commercialised, because no one would, was to make GLP compliant. So it's an exemption in the TGA to say that radiopharmaceutical productions within hospitals can be good local production, doesn't have to be good manufacturing production. And that's a massive difference in cost of production.
Right. So obviously in the United States you have Lantheus and Telix, selling huge amounts of product.
Yeah.
Is that the same? Is that available in Australia?
So it's available in Australia, but the problem is without the requirement for a GMP compliant production, say in my facility I can produce gallium PSMA-11, it's an off-patent agent, so we can produce that for a fraction of the cost that would be required to buy a product that's GMP compliant. So it makes the market much harder in Australia for companies like Telix and Lantheus and Cyclotech and those companies, because people like myself will have radiochemists that can produce it in-house.
Right. And the agents that you're using in Australia, is that functionally the same? Is it more or less the same? Is it exactly the same?
Exactly.
What's the difference?
So the PSMA peptide is exactly the same. So the PSMA-11 off-patent agent developed in Germany about 2011, 2012, introduced into the clinic there and then here very, very fast, is the same. It's the labelling that has been patented in the US. So it's the way that they chemically put things together that has been patented, or whether you're using fluorine 18s. Gallium versus fluorine. The labelling itself is what's patented. The PSMA-11 itself is not patented. But, we are going to have changes in imaging. So we've got the Clarity product, copper-64 Biz PSMA. Now that is probably a bit of a leap in what is happening within the cell when we inject that product. The copper-64 Biz PSMA agent is very interesting because it's not just one peptide, it's actually two. And it's got a sarcophagene label or central core to it. So when that is injected, it enters the cell in a different way and it gets stuck in the cell, while the PSMA-11 and the DCFP-well tends to go in and out, which means that you can image over a longer period of time. You get higher uptake within the cell, and that's super interesting. Whether it translates to better patient outcomes? We don't know, but we do need to look at that.
Yeah. And is the reason for that, is it the size of the molecule, or is it the fact there's two of them? Why does it stay in the cell so much longer?
I don't know, but it does. It does stay in the cell for longer, and that isn't technically, it's an advantage when you're looking at imaging, because we know that with PSMA, it's like a little elevator. Basically, you on the cancer cell surface, you've got the peptide binds to the enzymatic domain of the receptor, and then gets pulled into the cell. Some get pulled into the cell, some don't get pulled into the cell, but you want them pulled into the cell, and then you want the receptor to regrow or extravasate, so that you've got it's there again and can then bind and pull into the cell again. So we know we get increased uptake over time. That will technically improve our sensitivity for PET scans. So, that's a good thing.
Right. So you're basically accumulating more of that kind of radioactive particles that you're imaging.
In the right space and clearing it out from everywhere else. One of the big problems with prostate imaging is that your bladder is right next to your prostate. Most of these agents are excreted renally, so you get this big hot balloon right next to where you're trying to see something very small.
Right.
So it's very, very difficult. And we know that with gallium PSMA-11, with DCF-PYL, these agents, we miss a lot of local recurrences because it just gets drowned out by the bladder activity, and the half-life of these agents is not long enough to get rid of that.
So what's the difference in half-life we're talking about, and how does that relevant?
Yeah, so gallium's a 1 hour half-life, fluorine's a 2 hour half-life, and copper-64 is a 12 hour half-life. So it's really different. Now we know we've got good sensitivity, we've really got great specificity with gallium and fluorine agents that we currently use in clinical practice. What we need to find out, and what trials are all about, is finding out whether, the longer half-life agents also have really good specificity as well as good sensitivity. And if we have, how much can we dramatically improve sensitivity? And quite often, accuracy is like a seesaw. If you improve one, you reduce the quality of the other. So you need to make sure you're not reducing this specificity too much, but your sensitivity can go up.
Right.
So that's what research is about. And that's why we need head-on comparison trials.
Yep.
So we're doing one of those at the moment between gallium PSMA-11 and copper-64 bis PSMA in men who have biochemical recurrence following radical prostatectomy. It's an important space. Those men have an opportunity for cure if you get it right. But about half the scans with gallium PSMA-11 at the moment, or DCF-PYL, at a stage at which men are curable, are still negative. So we don't actually,
so you're not seeing it.
We don't know where the cancer is, and that's the most sensitive technique we've got. So the question is, if we use a bifid agent with a longer half-life, do we increase detection? And does that, A, is that real, what we see, is it correct? And B, does it change management? And most importantly, does it actually change outcome for those patients?
Yeah.
So, at St Vincent's, that's what we're doing. We're doing these sort of head-on comparison trials because, for me, it's great to get registration for new agents, but how do I know as a clinician, or how do my colleagues know what we should use? We have to know which one's better than the other and in which situation.
Yep.
So that's why I like to compare them. And I think it's brave of companies to allow comparisons.
Because it's a risky moment for any company, right?
Oh, yeah, it's super risky. And I'm not going to, I love publishing stuff, so I certainly wouldn't keep it quiet if it worked or if it didn't work.
Yeah. And so just, there's so many strings we could pull on there. But just to rewind the clock a bit, so gallium versus fluorine, do you have an opinion on the difference of those? Are they functionally the same in terms of outcomes?
So there's publications to show that they're the same, basically. People can quibble over, a gallium versus fluorine, but so similar that you could use one or the other. It doesn't matter. It doesn't matter. There's one agent, it's a fluorine 18-1007, it's a PSMA 1007, which got really low activity in the bladder, but it's got this really nasty bone uptake that's benign. And very confusing. I mean, when you have a cancer that goes to bone and then an agent that causes all this activity in bone, it's not helpful. It's just not helpful. They shouldn't use it.
Right. Why don't we set the scene a bit? So a patient comes in with prostate cancer. What's the current treatment journey? And then where are you getting involved with these imaging agents?
From the beginning. So in fact, we've been doing a trial. We actually did a multi-centre trial across Australia called the primary trial. Say a patient comes in without prostate cancer and they have an MRI and it's equivocal. You can do a PSMA PET now, and it will tell you very accurately whether or not you need a biopsy. So we're doing a randomised trial across Australia called Primary 2. Where based on the PSMA PET scan, the patient either does or does not have a biopsy.
Right.
That'll be super interesting.
Pre-biopsy.
Pre-biopsy.
And then biopsies, are they risky? What's involved in
Well,
the
I
biopsy?
mean, I don't know. Do you want to have 30 cores, needles, under general anaesthetic up your ass if you actually don't need it?
No.
No, I mean, the fact is, when I went for the original grant for that, I just put up a picture I got from the internet, sort of, internet porn almost, of a man with a black mask over his, stirrups under general anaesthetic with 30 needles.
Terrifying. Terrifying.
So if you don't need that, and a lot of men get diagnosed with cancer that is not significant cancer. It's actually not cancer, but it's called cancer. It's low grade. It's probably never going to progress. They're never going to die from it. They don't need a radical. One of the things I really like about PSMA is it can eliminate the need for the biopsy. You never get the diagnosis of low cancer, and it picks up all of the really significant cancers. So it doesn't take over MR. It adds information to it. And so I think that we're really going to see PSMA moving earlier in the space as well as later. And then in terms of staging, once you've got a diagnosis of cancer, it's very good at picking up metastatic disease or something that's already spread out of the prostate. So it helps the clinician know whether it's futile to do the radical prostatectomy or the chances of needing salvage radiotherapy, hormone therapy, systemic therapy as well. What we haven't got good at in the journey of prostate cancer for patients is identifying whether patients are actually responding to treatment. We haven't really adequately developed the criteria. And what big companies haven't done with systemic therapies is embed PSMA PET into their prospective trials serially, so we can see whether we can use it instead of the old technology like CT and bone scan. So that's a frontier that we need to cross, but we don't have any data to do it.
Right. So it's interesting that you're doing it kind of before pre-diagnosis. So would this be like a wide screening tool, or is this you've come in presenting with some symptoms?
So no, this will be in patients who've had an MR because they've got a rising PSA or a family history or some reason why there's a suspicion of prostate cancer, right?
Right.
And the MR is there's two reasons why we potentially might use it in the future. If the MR is equivocal or negative but not normal, it's very useful in that situation in eliminating the possibility of significant cancer. Right. So those guys, quite often they'll go, we don't know what the MR is showing, but we're a bit worried, so we're going to do a biopsy. If the PSMA is negative, then we know from previous work that we've done that the chances of actually having clinically significant malignancy if the PSMA is negative as well is vanishingly small. So you can be comfortable with not having the biopsy. And then if you've got a PIRADS 4 or 5, so more aggressive findings on the MR, a lot of patients don't want a biopsy, they just want to have treatment, or maybe one needle, not 30. And PSMA PET's super good at that too. So this is a new emerging use of PSMA PET. Is something that we've been really working on in Australia, and that's because of our GLP compliant laws allow us to do a lot more PSMA PET than you can get in the US. Right. So I think we will establish whether or not we can do that. But it is a fun, interesting, and I think really helpful use of PSMA PET.
And so this is still at the research phase, or is this how you'd actually treat people that come in at St Vincent's?
So we do have people coming in clinically for this in Australia now, but it's at the research phase. So we know that we know how accurate it is in that setting. We know it looks like it's useful, and now we're doing randomised trials with and without PSMA PET with the MR to see whether in fact it is useful. And in fact, whether we miss anything significant.
Right. Okay, let's move on the patient journey then. So you have done the biopsy and it comes back, something's seen. What happens next? So
then they get what we call a staging PSMA PET scan. And we know from really great work that was done across Australia called the Pro-PSMA study that it's much better than conventional staging CT and bone scan for identification of metastatic disease. Right. So if you have metastatic disease identified on a PSMA PET scan, that needs treatment. If you don't see it, then you can have definitive primary therapy with good outcomes.
Right. And what's the primary therapy there?
So radical prostatectomy or radiotherapy.
Right.
And some patients do have lymph node dissections as well. What's interesting about PSMA PET is I think you alluded before the podcast about its sensitivity needing to be improved. And what you're talking about there is when we've got very, very small nodes in the pelvis, whether we can detect them accurately. And that's actually a limitation of the cameras. In the PET cameras themselves can only really detect down to about 4 millimeters. But we don't Cancer doesn't stop at 4 millimeters. Obviously, it goes from 1 cell up to 4 millimeters, up to 10 millimeters, up to 20 millimeters. So we can only detect down to a certain level. And that's given us, with gallium PSMA 11 and with fluorine 18, our sensitivity is between 25 and 40 to 50% for lymph node detection.
So that seems kind of low.
It's low. It's
and
really
so these are
low.
people that have it, but you're not seeing it.
We don't see it.
So they think they've got the all clear.
They will be told they've got the all clear, but when the lymph node, and it's because not everyone does lymph node dissections, so there will be lymph nodes that are missed on PSMA PET that are small. The question is how significant they are, and whether it changes their outcome or not. I think, so our specificity is very good, 95%, 96%.
So if it lights up, it's there.
If you see it, it's real. So the way to use that, or the way the clinicians are using that at the moment to say, well, we know we might miss small stuff, so let's assume if you've got high clinical risk, you might have small stuff there, and we're going to manage that. But if it's positive on the PSMA PET, we know it's there. So it helps if it's positive. If it's not positive, they go on clinical risk.
Right.
So we can improve sensitivity over that, and I'm sure we will. And then what we have to measure is whether that's worthwhile improvement.
Right. And what are the ways we can improve that sensitivity?
So better cameras would really help.
Is it really the camera? Is it the imaging agent? Is it what?
So it's both.
It's both.
So we use crystals, so very, very small cut crystals, so 2 millimeter size crystals that are bunched together, especially with digital cameras, to look. So that's how we get to the 2 by 2 millimeter voxel size in terms of detection of radiation. And then we also, we also have binding affinity differences with the peptides. So some peptides bind really, really strongly and continue to concentrate in the cell. You get a brighter signal. So the two things together, smaller crystals, better digital technology, and better binding affinity of the peptides themselves, all will act to improve our sensitivity. What we have to be careful of in these really, really sensitive cameras is sensitivity and specificity is a bit like a seesaw. I think I said that before. If you improve your sensitivity, sometimes your specificity tanks. So you have to, this is why trials are so important, because you have to prove that even though you see more, it's useful more, not useless more.
Yeah, exactly. So where were we at to? So you had a prostatectomy. What happens next? You're testing
hopefully
for
you're
recurrence.
cured, right?
Hopefully you're cured, yeah.
And most people are. About 50% of people will have biochemical recurrence following radical prostatectomy. Once again, PSMA PET, it's standard use in Australia now for that, and in the US, actually across the world now. It's great for biochemical.
Do you just keep coming back just in case it turns back?
Well, no. If people keep having it, then that's a problem, because we know, another study that we did, if you have a PSA of about 0.2, your chance of cure with biochemical recurrence with salvage radiotherapy is good. It's excellent. We know from work we've done at St Vincent's that if you have a negative scan, so couldn't see anything with a median PSA of 0.23, and those patients went for salvage radiotherapy without having seen anything, their cure rate at 3 years was 87%.
Right.
So it must have been in the fossa, and we were missing it, because treatment of just the prostate bed itself worked without extensive radiotherapy everywhere. What's unfortunately happening is people like to see things in order to treat them, especially surgeons. They don't want to admit that they may have failed locally. So they'll say, we'll just wait, we'll do another scan. But every 0.1 that the PSA rises, that patient's chance of cure goes down.
Right.
So chasing with multiple PETs to wait until it's positive, it's the wrong thing to do. So it's a mentality thing. We have to find, either we have to change mentality and treat when it's negative in the fossa, which doesn't look like it's going to happen. Right. Surgeons don't want to do that. Or we get more sensitive techniques that detect disease in the fossa so patients get more appropriately treated.
What's this term, fossa?
So the fossa is the surgical site. So when someone shells out your prostate, what's left is the fossa, basically.
And I guess the issue is that even a prostatectomy, it's like a radical surgery, isn't it?
It's a radical surgery.
Yeah.
It's really radical surgery. And different surgeons say what their side effects are going to be, but it is really radical surgery. Half the men will be impotent, half of them will be incontinent and have to wear continence pads for the rest of their lives or significant period of time after the surgery. It's not insignificant surgery.
Yeah, it's something that struck me when I first started looking into this space was how, what's the right word?
Morbid.
Like you're treated, but you have side effects.
Oh, really?
You have this impression that prostate cancer is usually treatable, but
that's why,
at a heavy cost.
That's why I love the idea of doing the primary study, because, this idea that you could eliminate the need for biopsy by eliminate the need for radical prostatectomy due to anxiety in patients with low-grade cancer by just not doing the biopsy, but still not being at risk. You know what I mean? If you can use a non-invasive imaging technique to eliminate the risk of significant cancer, then I think that's a really good thing.
So I understand you're working on a trial that's head-to-head Clarity versus gallium PSMA. Is that a Illuccix, or is that just a gallium PSMA agent?
No, it's just gallium PSMA 11 that we make, using GLP compliant production in our department. Definitely not Illuccix. And it is a head-to-head comparison in biochemical recurrence with men who have a rising PSA that is at a level that they're still curable.
How many patients is it?
50 patients. And, I think that's super fun. We're doing management impact after the gallium 11. So we're ringing the surgeon and saying, or the radiation oncologist, and saying, this is the result. What are you planning to do? And then we ring them again after the copper 64 bis PSMA, and we said, what are you going to do? And then afterwards we'll look and see what they actually did, because it does change. And then what happened to the patient? So did that patient, following treatment or following no treatment, have a better outcome or have a good outcome? Like, what percentage of patients were cured?
Do you think 50 patients is enough to get that? Or is this just a study that will form a body
So it's
data?
a power calculation detection rate, 50 patients to look at a 20% difference in management impact.
Right.
Primary endpoint's actually detection rate. But so yes, definitely. If we see in order to make a difference in terms of whether I choose to, when we first started, we were doing choline for a prostate, and PSMA 11 came along, and one of my surgeons rang me and said, I want to move to this new thing. It's like, no. There's no publications. There's something in a mouse. I'm not moving. Prove it. So he gave me some money to do a head-on comparison trial. And we did the first 10 patients, and we biopsied everyone. And in every single patient, we detected more disease, more sites, more metastatic disease. And after 10 patients, and when we biopsied them, it was all true positive. So after 10 patients on PSMA, I had to close my clinical service for choline. We moved everyone over to PSMA, and that's where we've been since. So if I'm going to choose to move from PSMA 11 to, a bis PSMA or a different agent, I need to know that it's better, and I need to know that it's going to change outcomes.
I mean, there's absolute fortunes at stake here. You've got two $10 billion companies selling PSMA agents. What do you think it would take to change that practice in the United States and globally, or even in Australia? I mean, that's a question that investors are trying to grapple with as well.
Yeah, so it needs to change patient outcomes. The important thing is, if you're sitting here and you have biochemical recurrence and you do two agents and one identifies a lesion that's going to change your life, you want that one, don't you?
Absolutely.
I want that one, even though I don't have a prostate. But that's what I think should dictate the market, and that's what I think should dictate, what we should be using. Now, I'm an idealist, of course. I'm sure there's many other things that dictate that: access, ability to make things, ability to scale, all of that kind of stuff, price. But it needs to be that when you do a scan with one agent, patient outcomes are better than when you do a scan with another agent.
So do you think there's if you can prove that you can see smaller lesions and that leads to better outcomes, do you think the majority of physicians will switch to that better agent?
Yep.
They will.
I would. I've already done it once. Immediately. I closed my entire clinical service. It was massive. It was terrible. But I would do it again.
Terrible, just changing the way you work?
Yeah, I mean, we had to move from production of one to the production of another. We had to, it's just difficult doing that kind of stuff, getting access. So I do think that if you prove, I don't think sensitivity is enough, right? But if you can prove sensitivity without the loss of specificity, if you can prove that the improvement in sensitivity has an impact on that patient's outcome, then yes. And does one trial do that? No, but it has a big impact.
So I'm trying to understand. Another question is, PSMA, Telix has built a $10 billion business here, and it's enormous. We're chatting before, and you said there's like 100 PSMA agents. Can you help us understand, what Illuccix is?
There's probably not 100. How's that? There's probably maybe 15.
15. Anyway, a number of other agents. And funnily enough, Telix isn't used in Australia widely, as you're saying. Can Can you help elaborate on that? Like why, how did that state of the market end up the way it is now?
I think you could talk about the US. I mean, what Telix did is just stunning. They took a 1 hour half-life agent and managed to get it into hundreds of clinics in the US, GMP compliant, in a timely fashion for product. I mean, doing that, can you imagine that? Like
Half-life of 1 hour.
you know, you'd need thousands of Ferraris to in most places. So what they did was strategically incredible. They pulled off a feat. It's not the PSMA 11 that pulled off the feat. It's the ability to roll out and scale a 1 hour half-life agent that was the feat. So hats off to Telix. You know, Australia's a very different space, and we had been doing gallium PSMA 11 for quite a long time. So unless Australia changes its laws in GLP compliant production, then, you know, it's difficult. But we're not, I don't think, an important market. And the problem for Australia remains that our cardiac, brain, lung, bone, liver scans that are done in Australia, across Australia in hospitals for sick patients, are not commercially viable products. So if the law was changed in Australia to go from GLP to GMP compliant production mandatory, we wouldn't have most of those capabilities.
They just wouldn't come here. They wouldn't be...
No, I mean, we lose, you know, people decide just not to produce things in nuclear medicine because it's not commercially viable, which means that, oh, we're sorry, you know, you've got this rare cancer. We can't do anything for you because no one has produced the peptide even, or no one has produced the agent. And if you took that to another level by saying everything has to be GMP compliant, when there are no good facilities for GMP compliant production in Australia, it would lead to the collapse of nuclear medicine in Australia. America's had very different laws for a very long time. Canada's also got very different laws. Europe's got different laws. So applying what happens in the US and saying it should happen in Australia is easy to say and difficult to implement.
Is the GMP, is this process, is it mostly regulatory, would you say?
It's regulatory. A lot of it's regulatory. A lot of it's to do with the documentation of how it's done and whether it's done in clean rooms without any particulate matter. So, you know, I guess the question is whether GLP compliant production is safe. Certainly in Australia, in my many years as a nuclear medicine physician, I haven't seen any issues with production that's led to adverse patient outcomes from GLP compliant production. So it's the cost of the the bureaucratic requirements of GMP that is very, very, very difficult.
It's so interesting how these regulatory decisions and processes create these huge outcomes.
So it's you know, it's a double-edged sword, isn't it? We don't have GMP compliant capability and or you know, sort of viability in Australia, in my opinion, particularly for imaging. But that's also led to innovation. I could never have done these head-on comparison trials and these imaging trials as primary 2 randomised trial across Australia using GMP compliant production of PSMA PET at $750 a pop. We couldn't do the biochemical outcome papers that we did were all dependent on GLP compliant PSMA 11 production. The head-on comparison trial I'm doing at the moment is, you know, the cost is outrageous in the US for doing anything like this because you're paying $5,000 to $15,000 a scan.
Yeah, it makes a difference.
Massive.
So just while we stay on that trial for a little bit longer, so it's 4 millimetre, what would you call them?
4 millimetre deposits that we detect on PET scans.
And
that's a capability. Well, I'd love to. So the brighter it is, the easier it is to see on a PET scan. Previous study that we did, we had a 95% sensitivity for 5 millimetre deposits and a 0% sensitivity for 2 millimetre deposits.
Didn't see any at all.
None. So we've upgraded our cameras since then. We have nice digital PET, prolonged imaging. So, you know, I would like us to get down routinely to picking up 1 to 2 millimetre deposits. That would be cool. If we use a longer half-life agent that concentrates better in the cell, doesn't wash out, but whether we will, you know, so then it comes down to improving sensitivity by 1 millimetre or 2 millimetres, does that change your patient outcome?
And I guess the hope here is that Clarity's agent is 12-hour half-life versus 1. So how would that affect the cell? Could you just
because it continues to concentrate in the cell, technically, the sensitivity should be better. And you can see from Clarity's first papers, so they put out Propeller and Cobra already. their phase 1 trial. That had gallium PSMA 11 in it, and we know that the concentration within the big tumour deposits within the prostate were about 3 times that of gallium PSMA 11. And we also know from the COBRA trial that their 24-hour images have beautiful detection of lymph nodes, but it wasn't a head-on comparison trial, so you can't see what we would have seen on gallium. I think just 1 or 2 patients had it. So the question is, how different is that?
Right. And how do you know how sensitive it is at the end if you can't see anything below a certain size?
You don't. I mean, so you have to do lymph node dissections to get accuracy. Accuracy is very difficult. So you have to, you can't do autopsies on people to see whether they were positive or not. You don't want to do big lymph node dissections on people and cut stuff out. It's too invasive. So you have to rely on detection rate. You have to rely on response to therapy, and you have to rely on sort of proxy measures. It's quite difficult, actually.
Yeah. You mentioned some crazy number of the trials that you're currently working on and have worked on. So how many trials are you working on at the moment?
Oh, a lot. So we're doing a lot of imaging trials. I think imaging trials are super important. And we're doing that predominantly with PSMA PET. But then we're also doing a lot of therapy trials. So we label, at the moment, it's super fun if you're a science geek, in our department, where we use lutetium, we use actinium, we use copper-64, copper-67, we use gallium, we use fluorine. We're going to be using lead-212.
So you're using all of these agents?
All these, they're elements, actually. They're not agents, they're elements. And then we can clip them onto any kind of targets. So, the targets that we're using are increasing as well. We use GRPR, so bombesin as well, PSMA, and then there are other targets that are coming along like KLK3, STEAP1, STEAP2. We're going to be labelling more things to more things, and we're going to be adding more elements that are radioactive that have different capabilities. So at the moment we're using lutetium, it's a beta emitter. We use actinium as well, and we do that particularly to salvage patients who failed lutetium therapy in the late prostate cancer space. Lead is another alpha agent that's looking super interesting with a really short half-life, and I think that will be hopefully clinically very effective, but it's in phase 1 at the moment. Copper-67 is another beta emitter that, Clarity are labelling to their sarcophageine with the bifid agent. That could be super good. We need to see the data from their phase 1. And then...
so you combine that with a lead?
We combine that with... No, you combine that. So what you have is your radionuclide, your warhead, then you have your chelator, which is the chemistry, and then your peptide. So say you've got the copper-67, the radionuclide, it's a beta emitter, then you can label that to PSMA, you can label that to GRPR, you can label that to DOTA, and then it will attack whichever cancer has that receptor on it. So you can clip and unclip the peptide, and you can clip and unclip the radionuclide. And the radionuclide you use, it's become really important, because with lead, really short half-life, so you have to have a peptide that's going to get into the cell really fast, before it decays, because you don't want it to decay elsewhere. Similarly, if you have an antibody, antibodies take about 5 days to get into the cell, so you have to have a long half-life like actinium and things. So what you clip onto what is really important, and how effective it's going to be, and how toxic it's going to be.
And how have you found working with actinium? Is that more toxic? It's obviously very different to a beta emitter.
Yeah, so I have loved using actinium PSMA 617 in our patients on trial. It's a Novartis trial called the ACTION trial, so phase 1 dose escalation. They're moving into randomised phase 2-3 trials with PSMA ACTION, which is salvaging patients following lutetium PSMA.
This is right at the end of their
Right at the
treatment cycle.
end. So when these patients don't have anything left, it's very disappointing for these patients when they get lutetium PSMA and it's not working.
And how effective is that? How many people are in that situation?
So it's about 3,000 patients die from prostate cancer in Australia every year. Most of those patients would be eligible at some stage for lutetium PSMA. Lutetium PSMA only works for a period of time, and about half the patients who receive lutetium don't have a good enough response, and so they need intensification. So the question is, what can we intensify with, and how can we help non-responders? And then the other question is, how can we take patients who are receiving radionuclide therapy for metastatic prostate cancer and make it even better? And I think, PSMAs are targeted, or anything labelled to PSMA is a targeted therapy. I think targeted therapy is really good, but then we need to think about what other targeted therapies will actually enhance its effect in the cell. And we've just published in the Lancet Oncology about 3 weeks ago looking at the combination of lutetium, PSMA 617, to enzalutamide, which is a hormone blocker. So targeting the androgen receptor and targeting the PSMA receptor together is better than just targeting one receptor.
What did you find? How much better?
Oh, much better. So we had an 8-month overall survival benefit in these men who have a 5% 5-year survival. So abysmal 5-year survival in these patients. That is the longest overall survival benefit that's ever been published in metastatic castrate-resistant prostate cancer for any agent. So, I think what's really nice about where we're moving in prostate cancer is we're moving to targeted therapies. I would like to say we should be moving away from broad systemic poisons. You can tell I'm not an oncologist. So instead of just poisoning generally, we're going for targets. And the way I think we should be going is for using multiple targets at once and then using biomarkers to guide which targets we should be using in which patient. And we're going to get really great outcomes once we, figure out how to do that.
Yeah, and I imagine there's a trade-off between, the beta emitters and the alpha emitters, actinium and lead versus lutetium,
an alpha technically will be more toxic because it's more of a radionuclide, more of a warhead. So what we have to do then the holy grail is using the alpha to get really good cell kill in the cancer, but not affecting what we call non-target organs. So, at the moment with actinium, PSMA 617, it's pretty toxic to the salivary glands because the salivary glands take up PSMA and the kidneys also take up PSMA. So we need to look for peptides that focus in the cancer but don't affect salivary glands or kidneys.
Do you think those actinium and lead, they'll move further up the line of treatment, or they'll always stay as the... No, they're definitely
going to move. They're going to move. No, super looking forward to it moving from, we've shown with using the combination treatment in metastatic castrate-resistant prostate cancer that we improved overall survival dramatically. What if we took it one step earlier and when patients were first diagnosed with metastatic hormone-sensitive prostate cancer, what if we're using it there with a non-toxic peptide that's effective at killing off aggressive cancer clones in conjunction with the androgen receptor also being targeted?
Pre-prostatectomy? Or would that be post?
Well, so some patients turn up, about 10%, 5% of patients turn up with metastatic disease at the time that they're diagnosed. It's called M1. Those patients never have a radical because it's already gone, it's already bolted. So in those patients and in patients who have a radical prostatectomy and then the disease comes back pretty badly, they're the ones who we're talking about. We call them metastatic hormone-sensitive prostate cancer. And I'd love to see the radionuclide space move there. But in order to do that, we have to have non-toxic, effective agents, and we have to team them up with other targeted therapies.
So which ones are you most excited about? Which ones do you think will fit those characteristics?
To tell the truth, I'm a real trialist, and I want to see how they function in trials. So I think that lead 212 is looking really exciting. It's got a short half-life. It's an alpha. It should be highly effective, but it has to be teamed with a non-toxic. You can't have salivary gland or kidney activity. Copper-67, similar, looks really effective in what they've reported from the phase 1, but you have to have non-toxic if you're going to move it very, very early in the space. Actinium the same, and lutetium. I still love lutetium. We managed to put lutetium with enzalutamide in the NSP trial together, and the toxicity went down with the addition of lutetium to enzalutamide. So we got lower toxicity with 2 drugs than we did with 1.
Why? How did that happen?
Because everyone did so well. So if you get rid of cancer in a patient, they feel great. So just eliminating cancer more effectively reduces side effects.
Right. Less treatment.
Less treatment.
Down as long.
Yeah. So fatigue went down, pain went down, health-related quality of life went down, health-related quality of life went up, physical activity went up. So, to me that's the holy grail. Everything we're doing, all this money people are spending in this industry has to come down to people feeling better and living longer.
So what are you most excited about then in these upcoming trials?
Making people feel better and live longer. I love this sort of magpie approach of, what are we going to combine at a cellular level? How can we optimize what we're doing to get better outcomes and which combination. They did it in lymphoma. They absolutely smashed the field in lymphoma by using 4 agents together, and they went from about a 5-year survival rate to a 5% 5-year survival rate to an 85% 5-year survival rate by using this combination technology. And that's where we will go in the next 5 years, I hope.
Right. And I guess on the imaging side, what are you most excited about there coming up?
So on the imaging side, we do need better agents. So I do think we need to improve sensitivity while maintaining specificity. So that's agents that have better binding affinity in the cell. That's the important thing. One thing we haven't addressed at all, and I really want companies to start doing, is to embedding any kind of PSMA PET into their prospective trials in metastatic disease, because I really want to use it to look at treatment response. I think it'd be very, very good. And at the moment, we use CT and bone scan, which is just archaic technology. It's very inaccurate. But because no companies ever want to pay, because it's so expensive in the US and Europe to do a PSMA PET, they don't put them into their trials. So we don't know how effective they are in predicting whether a patient's responding or not to treatment. So I want them to do that.
It's quite interesting how that regulation kind of limits what can be done in a trial if you have these agents that image better, but that just makes it cost prohibitive to include.
Cost prohibitive. And so, CT and bone scan, dead easy to do, dirt cheap,
What's the sensitivity
but not
on
accurate.
that?
It's terrible. It's terrible, but that's still what we're using in patients with metastatic disease to guide their treatment. Because we don't have the evidence from the PSMA PET in the metastatic disease setting to do that. And that's because companies haven't wanted to invest in their trials. See, you have a prospective trial. Everyone uses CT and bone scan because that's what the FDA requires. Then you'd have to put PSMA PET on top of that. And that's there's no advantage to the company to do that if you're a therapy company. But at the same time, to the community, we need to do that.
Well, that's a challenge, isn't it? Because you also need to get clinicians on board. Because it seems like there's a lot of options in these.
We're on board. We're on board. And, basically, as long as you're using the same PSMA peptide with each imaging time point, it doesn't matter what you use. We haven't shown, to date, no one's really shown big differences in the PSMA peptides that we've got. But that's why we have to do the head-on comparison studies.
You mean the existing agents are more or less the same? There isn't one that's
pretty well
better.
the same. You can choose one, choose the other. It's not very different.
Interesting. So how long will it take for this field to evolve to the state that you're seeing?
Well, I guess, what am I talking? We've been talking today about lots of different things, right? And that's sort of, 5, 6, 7 years worth of work. So I'd say 5, 6, 7 years, we're going to be in a very different space to where we are now.
Hopefully better imaging agents.
Better therapy agents, better combinations, better evidence from trials. And hopefully in Australia we'll even have some reimbursement for something for our patients.
So it's not reimbursed at all at the moment?
So the imaging is, but the therapy is not reimbursed at all. So if a patient has metastatic castrate-resistant prostate cancer and they want to access radionuclide therapy, they can either try and get on a trial, but there are very few trials open at the moment, or they can pay for it. And that cost at the moment is somewhere between $6,000 and $50,000.
It's like Novartis Pluvicto. Do you have to pay for that? Is that what you're referring to?
So at the moment, Novartis have TGA approval, but no funding. And no funding application. You have to pay the full price for Pluvicto, and I don't know the exact price, but it's many tens of thousands.
Tens of thousands
per dose, which means it's about 300, probably about 300,000 for a full course.
Right. So if a patient wants that, they're going to have to pay for that out of pocket.
Yep.
Why is that?
Because the
evidence is strong, is that it works.
So in Australia, in order to get funding, you have to improve quality of life years. And the trials that have been done with Novartis have been against inactive comparators predominantly. So the Vision trial, lutetium PSMA versus standard of care, excluding chemotherapy. So they didn't allow chemotherapy in the standard of care arm. They just allowed sort of almost palliative options, which meant that from a quality of life years, when you compare it to the comparator arm, it didn't improve survival as such because it wasn't an active comparator. So the way Australia dictates whether or not something's improved quality of life is different to the way other countries do it. It's harder to get approvals here.
I guess the challenge then is, if Novartis can get a Pluvicto approved in the United States, why would they go through that extra set of trials that are probably much more difficult to prove just for the Australian market?
They won't.
And they haven't.
No. So we're in a pickle in Australia in terms of getting approval for things. And that's very disappointing. A lot of the really important work in the beginning was done by Australian investigators and Australian institutions, and Australian men went on those trials and randomised. It's really hard asking patients to randomise on a trial when you have another inferior product. In Enzapi, it was gut-wrenching.
Give me the treatment.
Yeah. So we did enzalutamide versus enzalutamide plus lutetium PSMA 617. We know those men had a high risk of early treatment failure on enzalutamide, and they did fail early. And they did really well with the addition of lutetium. And you have to continue to randomise the patients as you see that happening again and again and again. So hats off to Australian men for being on randomised trials to provide evidence for everyone else as to what we should be doing. We know patients on trials actually survive longer than patients that aren't on trials.
Just in general.
But it's a hard thing to watch, right? It's a really hard thing to watch, and it's hard for them.
So what did they actually show in Pluvicto that was better than palliative care as opposed to chemotherapy?
So the Vision trial was actually standard of care versus standard of care plus lutetium PSMA 617, and the standard of care was second-line hormone treatment, steroid, bone-sparing agents. They were light stuff. What was super interesting really for the Vision trial was in fact that those guys who were on second-line hormone treatment did much, much better than the guys who were on other standard of care with the lutetium. So in fact, Vision was a bit of a messy combination trial, and it's subsequently shown really quite significant improved overall survival for those patients who were on the combination of hormone treatment plus lutetium PSMA. So it's it was not compared to nothing, but compared to very little. And the PSM addition trial, which was taking it earlier, also a very nice trial, but it compared it to second-line ARPI. So it's a hormone sensor, so patients have already failed an ARPI and all they did was swap it over.
What's an ARPI?
So an ARPI is an androgen receptor pathway inhibitor, so enzalutamide, apalutamide, darolutamide, those kind of hormone blockers. If you swap them, you do get a short-term response. But it's only short. Swap. Say you've on darolutamide, you swap it to apalutamide, or enzalutamide, swap it to abiraterone, you'll get a short-term response. That was the comparator arm. So what did Oliver Sartor call it once? He called it the weak sister comparator arm. I take a bit of umbrage to that. He could have called it a weak sibling. But
why? I'm just trying to understand this a bit better. So why was that sufficient in the United States? Why did the FDA not ask for more?
I don't know.
Or why is the TGA...
I don't know what the difference. It's not TGA. So the TGA have approved Pluvicto. It's getting it through PBAC or MSAC,
right,
or those approval agents for Medicare. Or for, approval funded by the government. So the government's got a much higher bar in Australia than in the US.
So how should have that Pluvicto trial been run? Or what would the government have needed to see? They'd need to see it versus chemotherapy versus...
An active comparator arm. So chemotherapy, so cabazitaxel chemotherapy. So that was done with therapy. So we did that in Australia. We did lutetium PSMA, and ANZUP Cancer Trials did that, and lutetium PSMA 617 monotherapy versus cabazitaxel chemotherapy, head-on comparison. And it dramatically improved depth of response and quality of life, the lutetium PSMA 617, but it didn't improve overall survival. So we didn't get evidence for that either.
And do you think that is like the reality, or do you think it was a small trial? Or
I think it's the reality. I mean, the fact is, and I've talked about using multiple targets. The reason I like using multiple targets, say you get cancer and we take out one of your tumour deposits and we look at it under a microscope, we know, because people have done that you never have PSMA receptor expression in every cell. Maybe only half of them, maybe 3/4 of them, and every deposit will be different. So using monotherapy with these agents, even if they're highly effective, will not kill every cell. So if we want to make a big hit on survival, you need to attack more than one receptor, basically. And until we do that effectively, we're not going to get dramatic overall survival benefit from theranostics or any other targeted treatment, because it's not expressed in every receptor, every cancer cell. So I think it's super good therapy. I think we're going to be absolutely making a difference in the future. No, PSMA as a target,
right,
And with all of these different agents that we can use, we're going to get better depth of response, better cell kill, we're going to get better treatment responses. But until we actually use them in combination, we're not going to get overall survival benefit.
Right.
Not
So
big
what do you
ones.
recommend to your patients in that final stage when they have these?
Well, I did the NSP trial, so that's androgen receptor pathway inhibitor plus lutetium,
Right.
or plus actinium, or plus copper, or plus lead. So I think they're two great agents together, right? Because you're different clonal populations, different targets, and you're really deepening your response and prolonging it. And we showed overall survival benefit as well. So I think to me, that's a given.
Do patients have access to that, or is that?
They do, but
Does
it's,
it have to
in
be in a
Australia.
trial, or
No,
how
there's
does it work?
absolutely, there's access to that.
There is access.
But, most of the people have it early. So this first line hormone treatment, we've now moved to the earlier space. And so the question is now, does that mean that radionuclide therapy should also move to the earlier space? And then later, should we combine it with something else? So I think what we combine with what is the work of the next 5 years, plus how we optimize cell kill by which target we use, which peptide has got the best binding affinity and the best sort of non-salivary gland, non-kidney toxicity, and then which radionuclide is going to be optimal. So that's how many combinations of things have I just said? There's just massive, actually.
Yeah, that's a lot of work.
It's a lot of work.
But I guess the good news is maybe some of these treatments aren't actually competing, and they could be used.
They're not going to be competing. I think it'll become quite clear when we should use what. Some things are going to be great early. Some things are going to be great late. The combination we use late is going to be very different to the combination that we use early. Yeah. And I actually think there's multiple bites to the cherry with this. So you get a patient on their journey. I'm not going to say they should have it before it's metastatic, that might happen in the long term. But you get a patient on a journey with aggressive prostate cancer. When it first becomes metastatic, do I think we should be using radionuclide therapy? We should be looking at it. Then when they progress later, again, because it's a lethal disease, and hopefully we can turn it into a really chronic disease rather than a rapidly lethal disease. I guess that's the goal. When they have it again later, should they be treated with the same thing, or do they need something different? Probably they need something different, and they probably need a different combination. So what we use when, how, and what with is the big question.
You mentioned bombesin. Can you talk about that, what it is, what you're targeting, where it would be helpful?
Yeah, so bombesin is a really interesting receptor as well. It's also expressed in prostate cancer, but I particularly like it in breast cancer.
Breast cancer.
So it's good in ER/PR positive breast cancer, and there's some subtypes of breast cancer, like a lobular subtype, where it's really hard to detect. So you do conventional imaging, CT and bone scan, they're usually negative, and these people have very vague symptoms. It's hard to find the cancer. There's nothing that lights up, and so these patients have a delayed treatment or delayed diagnosis of metastatic disease, because often sits around the bowel or in the peritoneum or in weird spaces. So we've just done a trial in women with ER/PR positive HER2 negative breast cancer and at risk of metastatic disease, and it was really nice. Bombesin.
What did it show?
So 75% of patients had positive findings on the bombesin compared to 50% of patients on FDG.
Right.
So it had a better detection rate than standard FDG, which is rebated in Australia for staging of these patients.
So there's an extra 25% of patients who saw something, they can be treated?
Yeah, so we need to, early studies. So that's the second trial we've done with ER/PR positive HER2 negative breast cancer. We did it with a copper bombesin agent, and we've done it now with gallium NeoB, which is a Novartis agent, so a Clarity agent, and they both showed benefit. So I do think there is definitely a role for bombesin in breast cancer that I would like to explore more, because we really don't have enough agents in the breast cancer space, and bombesin looks like a good candidate. And then bombesin in prostate cancer, we've also done a trial, and it looked interesting in detecting patients who are negative on PSMA PET, but I'm not sure how to use it. I find it a bit confusing.
Would it be, well, I guess, is it a case you're negative and then you have this extra test to see if there's anything lights up? Is that how it would be used? Or is it
maybe, but
why would it be difficult?
Once again, you have to prove benefit to the patient.
Benefit.
So, and those studies haven't been done.
Right.
So if you do PSMA PET and it's negative, and then you do a bomb% PET and it's positive, you A, have to know that it's true positive, and then B, you have to know that it's going to change patient management. And then you have to know that it changes patient management appropriately so that the outcome's better. Know that none of those trials have been done that I've seen.
I guess they're expensive, large trials probably to prove that final outcome.
They're complex to do, and you have to be invested in it, like co-PSMA, head-on comparison trial, management impact, outcome. They're good to do, but you need to be a bit of a nut.
So how are you actually working across all these trials and seeing patients, and how does it work at St. Vincent's?
It's just insane.
Yeah, how are you spending your time? Is there a large team? Like, how are you even managing all this?
We're actually a really small team. But we're a super functional team at St. Vincent's. Good at trials. I've grown up with trials, and really committed, and they enjoy it. So it's great to work with a team like that. We do need more resources. We absolutely always need more resources, or perhaps I need to do less.
Don't do that.
Do less.
Yeah.
But that's
5
the
years,
problem,
that's a long
right?
time. Speed it up.
And Australia's a great space to do trials. And companies want to come here, and they want to do trials here because we have a good regulatory environment for trials.
It sounds like it's a bit cheaper if you don't have to pay.
It depends whether we use GLP or GMP compliant production, and whether companies are happy with that. A lot of companies are not happy with that. And it depends whether the TGA or yes, maybe. Yeah. So it's quite complex. What I want Australia to be is a place though where we do the trials. Our patients go on trials, but they benefit from reimbursement ultimately. And that's not a space we're at the moment. We're at a space where we're developing the evidence for everyone else. Everyone else has got access and we do not.
What do you mean everybody else? Everybody else offshore? Or
Europe, US, multiple countries now have reimbursement. We do not have reimbursement. We don't have GLP compliant reimbursement for any radionuclide therapy, or we don't have a Medicare, we don't have PBAC for any radionuclide therapy at the moment, apart from iodine, which is about 70 years old. So, we really need a space for improvement, reimbursement. We've got great biotech in this space. We're doing great trials, but we need to get that to our patients.
Why is the reimbursement such an obstacle in Australia?
Because of the methods of reimbursement and the quality of life assessments and the health economics that is required are very, very stringent in Australia.
Is that because the amount of testing you have to do, like the size of the trial?
No, it's about improving. It's about the design of the trial. If you don't have the design of trial to show overall survival benefit in the US, but you do have to here, no one's going to do it just for here.
Right. Interesting.
And then,
it seems like a structural issue.
And then it's a hard leap. Yeah, I would love to see us having this discussion about how we can talk to government and companies about developing reimbursement pathways for theranostics in Australia for our patients.
It sounds like from the economic situation you've described, it almost needs to change. It's almost the regulations that need to change because the economics aren't. It's not like Australia's going to be bigger.
I think it's the way it's evaluated needs to change. I mean, obviously they get regulatory approval and funding in the US and Europe. Why with these trials are we not getting it here? Why is it so difficult here?
I mean, this translates to people not being treated, right?
Not being treated.
Getting a worse treatment or
dying,
not being limited from things.
Dying more painful deaths, not having the opportunity to live a couple more years. No, it's with a non-toxic agent, choosing to have chemotherapy. Once again, I'm an oncologist. I'm an oncologist. Probably wouldn't talk about chemo, but they lose their hair, they become anemic, they get sepsis, they go to intensive care. They're fatigued.
This is prostate cancer.
This is chemotherapy with prostate cancer. Like some people manage it, some people really don't tolerate chemotherapy. Lutetium PSMA, we've got 2 at the moment. We've got INT 617, are both used in Australia. They're non-toxic, so we get mild salivary gland, they get mild dry mouth. The hit on the marrow is very minor. Some people get a bit of nausea. It's great. So I want that. I really do.
There's a lot about AFR articles recently, or financial press articles about compounding and things like that. Do you have a take on that?
Well, I think I was in that AFR article, so I obviously do. And I think I said it before. We have GLP compliant production in Australia. If they shut down GLP compliant production in Australia, then we lose our entire nuclear medicine community. It's gone. It's dead. You won't be able to get a lung scan or a bone scan or a brain scan or any of those things. So that's a big ask And it's why I was on that newspaper article, because you can't request that without understanding fully the implications. And then, whether or not we've been using lutetium PSMA INT off patent for years at a lower cost to our patients, but obviously not reimbursed. That's been approved for Medicare funding by the government from the 1st of July. But now there's a patent challenge with Novartis, and, whether or not that's going to come through as funded treatment for our patients, I don't know. But I really want all involved parties to talk and come up with a solution for our patients.
Yeah.
I don't care what the solution is. I don't care what I inject, but I care about the fact that we don't have access.
Is there anything else you're working on that we haven't discussed? I think also there's a lot of high net worth and family offices that will be listening to this. Are there any things that you would be doing if you had a bit more funding or a bit of access to something?
God, I don't think I can do any more. But I really love what we're doing. I think I would be working more on combinations.
More combination work.
More combination work, yeah.
Well, it sounds like you got some recent success in doing just that.
Yeah, and, we've cemented in 2. Maybe we need 4. Like what radiation sensitizers are really good. How do we overcome radiation resistance? But even just, look, you can poke a finger in theranostics at the moment and you have a question that needs to be answered. So I love being involved with the trials. I love being involved with the companies. Most of what we're working on in the future is IP, right? It's not my IP. It's their IP. But we, as clinicians, we need to be involved in this to help try and guide companies to get better outcomes, right? Because it's not all commercial. Ultimately, good commercial outcomes are related to good patient outcomes.
Right.
Or they should be.
In terms of working with these companies, how have you found that? You're obviously like patient first and actually treating the patients. And then you have a number of companies trying to develop new treatments.
To tell the truth, I think the biotechs in Australia are fantastic, all of them. I think they're all great. And I don't know, I don't think there's a big difference. Industry, ultimately, if you're going to produce a product that's going to be worth a lot, it's because it's working. You have to prove it works. I want the same thing. I want something that works.
Don't we all?
Everyone does, right? That's what you want.
Yeah.
And so I think the best way to do that is by academia and industry working together.
And yeah, it's clearly amazing work you're doing. So thanks again.
Thank you.